first clone 1 is there that has this much DNA, followed by this DNA, followed by this DNA. You already have labeled the clones, like clone 1, clone 1 has gene A, clone 2 has gene B, clone 3 has C. You have already put them in order based on their occurrence on the DNA. That is called as an ordered library. That means to generate a ordered library from a random genomic library, one should have done chromosome walking like we have done in the previous slides. Okay, so you should have performed chromosome walking, and then you know which clone is has the fragment after which clone, and that will determine the gene order on the chromosome. If you know that, if you have put together a library like that, that library is called as ordered library. It's all right. So in today's class, I actually wanted to talk about differential screening. Differential screening is something similar to that of subtractive libraries that we have made. I want to find genes expressed in A, but not expressed in B. Okay, so something like that is what is called as differential screening. You are trying to find out genes that are specifically or A specific. Here I'm calling A, organism A, genes that are expressed only in A but not in B, something like that. So here is a library. People, you hear as per the question, this is a library. So that means you would have taken the genome from an organism and you have made a library in something like an E. coli. Okay, and now you have a library. Library is plated like this. The objective is to find out, it's usually cDNA. Okay, the objective is to find out the genes that are expressed in A alone. And if you take, for example, on this side, you just look on the focus on this part. Okay, you take RNA and then you convert it into cDNA library, and then you do colony hybridization like technique. Whichever colonies have given positive signals are expressed in A. Yes, that is pretty straightforward and simple. You can also have sample B, and I mean sample B, or it could be a treated, untreated, like that. In a different RNA sample, you have constructed labeled cDNA library, and you have done colony hybridization on the different second filter that you have. If you have signal on both the plates, like this colony that I put it encircled, okay, that means that particular gene is expressed in both A and B. If a colony is, say, for example, this one, which is not expressed here, that means it is expressed only in B but not A. If you have, say, for example, this one also gave a signal here, but not here, that means that particular gene is expressed only in A but not in B. People use these kinds of differential screening for several purposes. For example, people take the same type of cells, say XYZ, and they make it into two partitions, and one is treated with a compound, and the other one is untreated. And then they take RNA from both and they perform similar assay differential screening. So they will, untreated ones, they will try to find out what are the specific genes that are expressed only because of treated ones. then they know how the compound is likely to work. Some people also do it like this. They do it at the egg stage, and then say if the zygote, if this is a zygote, assume, and then it forms a blastocyst, and then a tadpole, or any of those things. Okay, a growth stage. You can isolate RNA from different cell stages, and then check which genes are expressed in these different stages. Okay, so differential screening is about trying to identify, screening a library to find out the genes that are expressed in a certain growth stage or a treatment protocol, and so on. So you'll only now you will be able to one will be able to identify the genes that are expressed in that particular stage. In this situation, in these kinds of experiments, people have to take care of something like this. The host that you take here should not have any similarity with the genome that you are testing for. Say you take the DNA, and then you make it fragmented, and then you make it restriction digestion, then you clone it into a vector like that, and then you put it into a host. If one is trying to do any assay like this, okay, you should not have that much homology between the DNA of the organism from which on which of which the library is being made to the host. If there is the same gene XYZ here and also XYZ here, right? What is the risk? If I'm trying to see for XYZ, all colonies will give a signal. Why is that? Because the host itself has that gene. So typically, what people would do is to, if this is an eukaryote, okay, and then they would choose a bacterial one. If it is fungi, it could be a fungi, humans, or anything, and the host is usually E. coli. So that there will be nucleotide differences between the genome on which the library is made and the host that is used to make the genome library. Okay. This will be the last one, and there is conceptually something very important. Until now, we have used probes that are only nucleic acids, right? Here are some of the probes. There are also probes that you can use to study at the level of proteins by using antibodies. I hope you already know what is IgG, right? Immunoglobulin G. And this is a kind of sandwich kind of method. I will not discuss that here, but I want to go into something directly application. There are two kinds of fusions that you should understand. Okay, fusion is where we have fusion is there are two different types. One is transcriptional fusion, and the other one is called as translational fusion. If I am using antibodies, then I'm actually trying to figure out proteins, not nucleic acids, right? So if I have a gene like this, and there is a promoter, and if I'm cloning the gene of interest here, okay, this is now one complete transcriptional unit. There is no promoter within this intergenic region. No promoter, no terminator. Anytime, say for example, if I put LAC-Z here, and this is PLAC or promoter for lacoperon, then anytime this gene, this promoter is activated, then LAC-Z will be produced. After that, whatever gene is there, that will be produced. So I can the gene of interest will also be expressed in that case. These are called as transcriptional fusions. Typically, what happens is that there is a vector. On the vector, you may have a small fragment of laxate here, and the promoter might be there, PLAC. If not any of these transcriptional fusions, and this will be the multiple cloning site. Within the multiple cloning site, you would cut and you ligate the gene of interest in this place. Then whenever if you induce this operon, you will get laxate or fragment truncated one, whatever may the case, followed by the production of this one. So it's just a kind of transcription. It's a kind of expression vector. Translational fusion is something little more than that, where you're actually fusing at the level of translation. That means assume that this is the laxate, and here is the promoter PLAC, and you are directly fusing the open reading frames of the laxate and the gene of interest. That means no promoter, no terminator, not even a stop codon. The start codon would have been here, ATG, I'm writing in DNA, and this will be a TGA. This would be one complete polypeptide. Half part or so is of laxate. Upon expression and upon transcription and translation, you would get a part of a protein, something like this, where half part of it is laxate and the other part is of the gene of interest. Okay, these are called as translational fusions. Okay, so why we are talking about it is we are going to use antibody immunochemical screening, and if you are going to use antibodies, there must be some expression of the gene, only that then the protein would have formed, and then we can use antibodies to identify the proteins. So here is it is a translational fusion. Okay, we are making a translational fusion. If you have read about zap, lambda zap, like in here, you would have identified it, that this is a translational fusion. That means, sorry, I forgot to mention here. So typically how it would be is you will have a vector and for translational fusion, okay? And this is the promoter, and here you'd have laxate or some other reporter it could be. And the multiple cloning site is here right after it. You would have cut it and inserted the gene of interest in frame. What does in frame mean? That means the reading frame would continue with that of what is continuing for laxate. The codons of laxate and the codons of the gene of interest should be read in one frame. That is called cloning in frame. Okay, so there is no stop codon in here. You only have stop codon here. When the mRNA is produced, it will be like this, and eventually it will result in a fusion protein like this one. So the multiple cloning site, or usually the cloning site, should be in frame. That is one care one should take. In the final assignment that we are going to do, you have to keep these things in mind, how you are cloning it. If you are cloning your gene, the gene of interest, and if you didn't put it in frame with the laxate here, then your answer could be wrong. Okay. So in this case, what people do is it can be used for anything, but typically it will be used for lambda phage vectors, and we will also discuss something about phage display. If we haven't discussed this, we have to discuss phage display. I think it's one of the questions in the assignment that I've given. If you have done it well, you would have already understood that. but otherwise we can discuss it some other time. So we make a library in lambda phage, lambda based vectors like lambda zep or lambda zt11, and here is the lag set, and it is in the polylinker or the multiple cloning site is within the reading frame of lag set. Anytime which restriction site you're choosing should depend on whether the gene of interest is going to be put in frame or not. And I also want to discuss one small thing. This is antigen, and antigen may have several epitopes. Epitope is the region where antibody recognizes. Say, for example, this is an epitope. If antibody is recognizing, I hope you also remember about the antibody structure in general. There are two disulfide bonds, two light chains and two heavy chains. There are disulfide bonds between these, something like this. And the antibody has these regions. They are the hypervariable regions, and they recognize, they have a region, complementary determining regions, which are the ones that interact with epitopes uh, of the antigens. Antigen um, Antigen could be a large protein molecule or other molecules, and they have several parts on their structures on their surface, which are usually the ones that are recognized, or the epitopes are recognized by the antibodies. Okay. When you say, I hope you also know about what is polyclonal and what is monoclonal. In monoclonal antibodies, all antibodies in that set recognize one epitope. Okay. Polyclonal is multiple epitopes, which might be on the same antigen or different antigens. Usually, yeah, different antigens. So we make this cDNA library, and the objective is to develop a fusion protein so that the gene of interest also will encode. This will be the region that I'm interested. This part may be a small part, may be cloned, translated along with the rest. So you make a cDNA library in lambda zap, for example, and then you plate it on the paper. I mean on the plate, and then you will put it for 4 to 6 hours at 37 degrees centigrade to obtain small plaques. You're not waiting until the whole thing is lysed or so on. Once small plaques, you're also incubating only for 4 to 6. You're not doing it for a longer time. Then you you put a nitrocellulose sheet on top of the plate, and you pick up and you overlay with uh, it already has IPTG. Why did we add IPTG? IPTG is isopropyl thiogalactosidase, thiogalactoside, which is the promoter, something like this. So the lag promoter is then activated, and then it will PLAC is now active, and it will start transcribing this and then translate it. Okay. So IPTG is the inducer. Once we add that and we wait for 4 hours, why are we adding? Because our gene of interest is put into lacoperon, or downstream of lac promoter. So the cells nearby would also start expressing, um, will start expressing the genes that are present in lacoperon. Sorry, not lacoperon. On the ZAP that we have constructed genomic library, and we waited for 4 hours, and now you take out the nitrocellulose sheet, and then you allow the adsorption. There will be some techniques like fixing the proteins onto the surface. Say if this is the nitrocellulose membrane, different plaques might be stuck onto this, and the proteins are exposed because this is anyway lytic phage. but then some cells would have expressed some of the lambda phage genes that are there, including the fusion protein that we are looking for. And now you will add antibodies. Antibodies are the ones that here you're adding antibodies for the specific protein that you want to find. So the antibody we may bind to one of the plaque regions, because you have retained your master plate. You know which plaque is actually having the phage of interest from the library, and you take that phage and then you will go for further characterization. Again, here screening is trying to find out the gene of interest within the library, and you're using a immunochemical. Immuno is basically you're using um, antibodies. And I forgot to mention the labeling method for this, right? That's why I put this picture and I didn't mention, probably. Sorry. So here, as we in the case of nucleic acids, we use radio-labeled phosphate, right? P32. Here we used iodine. Very easy way of trying to label iodine, immunoglobulins, or any proteins for that matter. You just treat it with iodine as a compound, radio-labeled iodine, and it adheres to the antibodies. Okay, and then when you add when you perform your assay, now these are basically these antibodies are already immunoglobulin or radio-labeled with iodine. Whereas in probes that we have made, we have to carry out an in vitro transcription or polymerization, again to incorporate the phosphate group. But here in labeling proteins, in this case, it's simple because you can directly add radio-labeled iodine to the already synthesized antibody. But there are also many other techniques, as we will see in blotting techniques, where they add an enzyme conjugate to this, which is little more, you have to do a chemical reaction for this thing. But otherwise, radio labeling is easy, but it is harmful. So people do not follow this as much unless very required. What we also have to remember is, for screening purposes, the probability of finding the clone or the plaque is actually difficult. It might be very few numbers of binding interaction between um, the protein, the antigen, and the antibody. In that case, we have to have highly sensitive ones, and for anywhere we need, the probability of binding is less, and we need a lot of, then we will need a lot of sensitivity. In those cases only, one will use radioactivity. Nowadays, you can think of it that radioactivity is kind of almost removed. We don't need to use as much of as radioactivity as people used to use in the olden days. Okay, I'll stop here, and if you have any questions, I would like to answer. Let me go back and see if I missed. Yeah, tonight I will post a question or so along with a video on chromosome walking. Okay, if there was if we had it in the classroom, it would have been actually a very nice one, but that's all right. I'll try to record a video and see if it works out well. Okay. Any questions? You can also ask about the assignment. I think I extended the deadline a little bit because of your vacation time, or I don't know what it is called. When is the next deadline?